But we wanted education that really supports the practical application of what our students are learning directly to the students' lives as well as for their professional work. And of high importance is to be able to offer affordable educational programs. So to support your health as well as your thirst for knowledge, Hawthorn offers 2 webinar presentations each month in holistic health and nutrition at no cost because we believe in your commitment to ongoing learning. Delighted that you've joined me for a few announcements, and then I've invited nutrition consultant Randy Friedman to be with us for our Book Nook segment. We'll review a new book and let you know what we think about it, because we think what we read matters too. And then at the top of the hour, we'll begin our presentation of Genomics, the science and application of genomic testing for nutrition professionals. And we have Drs. Joe Veltmann and Roberta Kline with us this evening from Genomic Solutions Now. I want to let everybody know that you're muted, and we do that for best sound quality. But we hope that you'll participate, and we're going to have a question and answer period at the end of the presentation. And so type your questions or your comments in at any point during the presentation. Don't have to wait until the end, and I'll pose them for you, for our presenters. And we'll do that at the end of the presentation. I want to let you know about a new course that we've got coming up. Hawthorn's Education Director, Janet Legwood, developed and teaches the Nutritional Needs for Healthy Aging course. And there's some good details about it. The course is open now for registration. So take a look at our website and find out what it's more about it. And just a reminder that Hawthorn does offer you our holistically focused webinars on the first and third Tuesday of each month. And at each webinar, at the end of it, we have a survey that we ask you to fill out. And it's an opportunity for you to give us feedback on the presentation that you'll just have reviewed and to let us know what you'd like to learn more about in upcoming webinars. So hope you'll take an opportunity to fill that out for us. And in our last webinar, it was on October 20th, we had herbalist Rosalie de la Forêt with us, and she had a great presentation. It was so beautiful. The slides itself were just so terrific. She's a fabulous teacher. The topic was staying healthy this season, top herbs for your immune system. Hope you'll check that out. It's archived up on our website now, and you'll be able to review that. And there's a lot of other previous presentations that should pique your interest. And if you did miss it, it's recorded. It's up there. We're recording this one too, and it'll be posted within just a few days. I think that's it for announcements. So let's go to our Book Nook segment. I'm here with Randy Friedman. Hi, Randy. I'm really looking forward to our presentation and so glad that you could join us a few minutes early.
Hi. Yes, thank you so much for having me.
All right. How's it feel to be a graduate, Randy?
It's great. I mean, gosh, it's been a little overwhelming. It seems like, but it's exciting. I actually miss all the schoolwork though. It's kind of, I didn't think I would, but I do. But I try to do a lot of outside studying as well. But I'm excited.
All right, congratulations.
Thank you.
Happy and proud. So we're going to talk about the book called The Hormone Secret, and it's written by Tammy Maraglia. And I'm just wondering what your impressions were of it, and if you really felt if the book would be helpful to you in your practice.
Yes. Well, I enjoyed reading it, and if you've read many other books on hormones, this one is broken down so it's easy to understand into like a 3-part approach that you can implement. There's not, some of the information is things you've heard before. It's not like there's anything really in here that's completely new information. But if you haven't read anything in the past about hormones, it's broken down really well. And then if you are pretty familiar with it, it's still, I believe you can still get a lot out of it because she does break it down into this 3-part approach.
Right, great. And my understanding it's presented as a 30-day plan to really promote it, to change the way you think, the way you move, the way you eat, the way you supplement. And there's a lot of plans out there and a lot on hormones lately. So I'm wondering what you thought that was interesting or unique with her writing style or with the approach that she uses in the 30-day plan.
Well, what I think I like best is that she breaks it down by each hormone and gives detailed lists of the foods, herbs, and supplements to support each one, and how they stimulate your body to manufacture and balance them naturally. And she feels testosterone is the missing link for optimal energy and wellness, which I don't usually hear much about, especially for women's hormones. I very rarely ever hear about testosterone, and she seems to think that that's the, the one to concentrate on. And she kind of covers it in a three-part approach. Her part 1 is like day 1 through 15, and she covers like the liver cleanse. And then part 2 is days 16 through 30, which is the adrenal healing. And then part 3 is days 16 through 30, and it's concurrently with the adrenal healing and the resetting of the hormones, so that she has it all build up to each other, and you kind of work it all together. And she covers things like healing, how to heal your adrenals, detoxifying your liver, different hormone-friendly foods, and she goes over all the herbs and supplements that boost each individual hormone. And then she really digs deep into testosterone. She really feels it's the missing link for optimal energy and wellness.
All right, really interesting. And there was a real section that I was real curious about. It really stood out. She raises a point about the correlation between micronutrients and hormone production. I wonder if that got your interest too.
Yeah, it did, because it seems to be micronutrients seem to be popping up more and more, and I've seen more and more books that based on a micronutrient diet, so it was interesting. And she felt that the micronutrients enable your body to produce the testosterone and the other hormones, which are vital to our health and our quality of life. And she feels that most people do not eat enough of the micronutrients. And she feels that the best place to find the best source for micronutrients is through food, and she feels it's really essential for providing the body the energy so it can go to work for you.
Right.
So
I really appreciate that a lot about her, is that she references food so much. It's like before you get into supplements and so many other product things like that. It's like bring it back to food. So there were some good recommendations, I think, for holistic approaches for diet and lifestyle suggestions. Can you talk about that a bit?
Yeah, so the second half of the book, so the whole first half of the book, she kind of goes through all the hormones, and she really sets it up by each hormone and going, really into detail with supplements and things like that. And then the second half of the book is where she covers parts 1 through 3 that I mentioned above. And she goes through like food schedules, foods to avoid, foods to include, and then she gives supplement recommendations and dosages. I really like the dosages, because sometimes people throw out the supplements, but they don't really tell you the dosage, and then you're kind of scrambling, trying to figure out, well, how much am I supposed to give for this? So I really like that. And she does address the bioidentical hormones, and then tests that you can run, and then she offers up a few recipes throughout the book too. So it's not a huge amount of recipes, but it's a small section of the book. Mm-hmm. And then she also discusses, like for lifestyle, she goes into belly breathing and meditation, and she covers the 5 steps of sleep and how they affect the adrenals and the rest and repair action of the body. And she also touches on how laughter, gratitude, and movement are so important. So she goes into a little bit of lifestyle education as well.
All right. Well, it'll be real interesting what our presenters have to say about this, if anything. So I'm looking forward to that. But what's your bottom line on this? Would you recommend the book?
Yeah, I definitely would. I mean, especially if you're interested in hormones, or if you're planning on working with people who on hormone programs. Because I feel you could take her 30-day program and implement it into your practice, and it gives you enough tools that you could create your own program to teach a workshop or a 30-day clinic, which I thought would be beneficial. You didn't kind of have to reinvent the wheel. Like I said, there's nothing earth-shattering in the book. There's nothing that I read and go, oh my God, I didn't know that. But again, I've read a lot of different things on hormones because it's a personal interest of mine. So, but either way, I found it very interesting, and I found, even with all the stuff that I've read before, I found it quite interesting. And I guess the biggest takeaway that I found that was different from the other books and articles that I've read is the part about the testosterone. Like I said, usually you think about testosterone for men and not so much for women, and she really focuses on it, and she refers to testosterone is the conductor of the orchestra, and that all the other hormones are the symphony. And so for her, she really feels testosterone it plays a vital role, which I have not really heard a lot. I have not read a lot about before. So I guess that's the biggest takeaway. And so I learned a lot more about testosterone than I had known before, especially for women.
You know, I think it's a good book. In my review of it, and you know, I'm impressed. You know, with your level where you're at right now, and your level of understanding and application, to be able to look at it and say there was nothing new, but you know, you could find the highlights and speak to it and really apply it. It just really speaks to your learning, Randy, and I appreciate that. Want to acknowledge it. And just one other thing here, just around providing dosages. I agree that that's not provided a lot, and I think there's good reason for it because everybody's unique, and you know, that's how we approach things. But to get a framework for, you know, what somebody's starting place or suggestions are, I think could be helpful, because a lot of times you're just in the dark about it, huh? Don't really know.
Well, exactly, and you know, people like us, we're more familiar with it. But if you were a person off the street reading this book, it'd be really helpful because you could go follow it yourself and pretty much help yourself. You know, if you weren't under a doctor's care, you at least have some sort of an idea where to start and what to do. Otherwise, you know, people just go out and start taking things. They don't really know how much they're supposed to take or what they're supposed to take.
Right. So that'd be good. You know, they can read the book and then work with you.
Yes.
There you go. All right. Well, thank you so much for taking the time and doing this. I'm glad to let people know about Tammy's book, and yeah, let us know what you think of it too. Thanks, Randy.
Okay, thank you.
You're welcome. And this is going to conclude our announcements and our Book Nook segment. And it's time for me to introduce our presenters and start the webinar. So let me get going with that. I know there's a number of you that have just tuned in, so I want to welcome you to Hawthorn University's webinar series, and thanks for joining us. I'm Paula Bartholomew. I get to be the moderator for the webinar, and we're going to learn more about genomics, the science and application of genomic testing for nutrition professionals, with Dr. Roberta Kline and Dr. Joe Veltmann. And as nutrition professionals, we're called upon to provide a plethora of information about diet and nutritional supplements, exercise regimens, and lifestyle choices. And providing these personalized recommendations can be challenging because everyone's unique. It's based on their genotype, and what might work for one person is not going to work for everyone. Our genotype, the genes that make up our DNA code and provide the blueprint for the proteins our bodies need to function, can be difficult to decipher when we rely solely on a person's phenotype, their traits, or their physical characteristics. So with the completion of the Human Genome Project and the advent of genomic testing, today there's a new tool in a nutrition professional's toolbox that'll help us to provide personalized programs to nutritionally support a client during a healing process, to use specific nutritional therapies to decrease the risk of chronic illness as part of the disease prevention program, to really help understand biochemistry behind our nutritional recommendations, and be part of the genomic revolution. I think part one of the presentation today is really going to focus on the science behind genomic testing and how best to interpret the results using some real-life examples from our presenters' clinical files. And then in part two, we're going to really focus more on the clinical aspect and the case studies, and go more into using these tests and actually marketing and having it be part of your practice. So let me give you a little bit of background on our presenters. Dr. Roberta Kline is a physician and author, teacher, and entrepreneur, and she's passionate about empowering others with the knowledge and the tools they need to create health of mind, body, and spirit. She's board certified OBGYN and has additional expertise in functional medicine, genomics, epigenetics, and multiple healing modalities, and actively shares her knowledge and vision with others through teaching, collaboration, and speaking. And, you know, this is really, I love hearing this because Dr. Roberta feels deeply that our health is intimately connected with how the foods we eat and the environments we create physically, emotionally, energetically, and spiritually speak to our genes. Dr. Kline's vision is a future in which the person again becomes the focus, and various conventional and alternative healthcare providers work together to create truly personalized health plans that support an individual's unique need that's based on their genes. And then Dr. Joe Veltmann is a scientist, healthcare practitioner, expert in genomic testing, and interpretation, is an author, teacher, and innovator. He's over 25 years of experience as a researcher and nutritional function and integrative medicine practitioner. Dr. Joe's made a career of translating lab results into practical clinical application for patients. And Dr. Bobby and Joe are co-founders of Genomic Solutions Now and Geneticist Center for Personalized Health, and are dedicated to transforming healthcare through teaching others how to create individualized health unique to each person's DNA. So with that, welcome, Doctors Bobby and Joe.
Hi, Paula. Thank you.
Hi, Paula. Thanks very much. Thank
Absolutely.
You for the
Absolutely.
And we're ready to get started.
I know you've got some great information, so I'm going to turn it over to you and let you get going.
Thank you.
Thanks.
All right.
For the first couple of slides, I'll be your moderator and presenting to you today some of the science and the application of genomic testing for healthcare professionals in dealing with their clients on a unique personal level. We call this personalized nutrition. And one of the things that I've done over the last couple of years is try to educate healthcare professionals about this topic, because what we're finding is that there's a steep learning curve for even clinicians who have had years and years of medical training as well as nutritional training. So one of the things that we're going to do today, at least in the learning objectives, and some of you who may have participated in the NAPA conference in Minneapolis, and I see some of your hands going up. No, I'm just joking. You'll hear a little bit of this topic and this information again. And I heard at a conference that I was attending this past weekend that the best way to educate people is to give them 70% of what they've already heard and 30% of what they haven't. And so the intent behind this particular Part 1 presentation is, first of all, to understand your client's genotype so that we can personalize nutrition, health, and wellness programs unique to that individual. But one of the things we have to do first is differentiate between genetics versus genomics. And we'll spend a few slides helping you get your arms around these two concepts. And then what we're going to do is look at how genomics is being used in medicine today. And we're going to use 4 different examples: the transmethylation and transsulfuration pathway, which includes MTHFR. There's a big, big, big focus on MTHFR, and we're going to expand that a little bit because the methylation and transsulfuration pathways include a whole lot of other enzymes and genes which encode for those enzymes in these 2 pathways. We're also going to look at coenzyme Q10, vitamin D metabolism, and balancing a patient's emotional health and mental health. I think for every healthcare professional that enters into a relationship with a patient or client, the goal is to personalize and customize healing modalities, therapies, and treatments. And oftentimes, we're stuck with looking at intake forms, family disease histories of patients or clients' signs and symptoms, blood data and how that might be correlated to the signs and symptoms or the family history. And if we get into trouble, usually the patient or client will come back to us and complain about a therapy, medication, or a botanical that might not be doing what it was supposed to do. So what our goal here is, we want to get away from the phenotype and focus on the genotypic uniquenesses of every particular client. You probably have seen this in one or more of your courses. It's basically looking at the inherited disease patterns within a family tree. And these circles and squares are used to help a practitioner see how the patterns of inherited diseases go from one generation to the next. And we know that gene mutations are a cornerstone of inherited diseases. And we have been taught to believe that one bad gene leads to a debilitating condition. That disease is passed on from one generation to the other, and that's because that mutation is in the ovum or the sperm. And there's a list of those kinds of diseases that are inherited. And when it all's said and done, what happens is that most healthcare professionals come away with this concept of medical determinism. That is, your genes are fixed, and you're fated to whatever disease or diseases you inherit from your parents. The Human Genome Project, which was completed in 2003, has turned that concept on its head. We now know that DNA consists of about 20,000 to 30,000 genes. There are about 3.5 billion chemical base pairs or nucleotides that make up the human DNA. And gene variants contribute to the diversity that exists in the human population. The Human Genome Project has become the catalyst for the omics revolution. And I've just listed a few of the topics and areas where omics is playing a role. You've all heard of the microbiome. We have the proteomics, metagenomics, pharmacogenomics, and we're going to be focusing on the nutrigenomics. We now know that gene variants account for about 80 to 90% of all the chronic diseases that occur in humans. Some of these gene variants are harmless, whereas others can predispose a person to certain diseases and debilitating health conditions associated with aging. And I share this new information with you. This is information that came from the 1,000 Genomes Project Consortium, which charted the human genetic variation across the globe by cataloging gene variances of 2,500 plus people across many different ethnicities: African American, East and South Asia, Europe, and the Americas. And the goal of this consortium was to understand how genomic variants contribute to human disease. And what they found, this project was completed last month, 88 million sites in the human genome vary among people. That's about 1 in every 30 nucleotide base pairs. 96% of those sites are associated with differences in single DNA building blocks called single nucleotide polymorphisms, or SNPs. 4%, 3.6 million sites, consisted of short insertions or deletions of that DNA sequence. And the study tried to identify how many sites are associated with the diversity that exists in the human population, and that amounted to 4.1 to 5 million sites. There's something else that came out of this 1,000 Genomes Project Consortium, and that is that there were structural variants, or SVs, which account for deletions, insertions, duplications, that is, extra DNA copies, and inversions, where DNA sequences are changing the orientation of the molecule. And what they found were that these structural variants disproportionately account for many of the specific traits or diseases that people are at risk for. And what the term that I'm trying to help you understand, which you may or may not have heard, is that we're no longer focused on single genes as it relates to someone's disease risk. We're focused on multiple genes and how they play together or how they not play together in a polygenic approach towards understanding the disease process. Just a quick review here of the Human Genome Project and how SNPs account for many of the predispositions that the humans are exposed to in their risk for certain diseases. We now know that genes have plasticity. Gene expression can be upregulated or downregulated depending on the environment, lifestyle choices, dietary selections, and or nutritional supplements. When a cell replicates, it can undergo many errors, and this can account for these polymorphisms. And when one nucleotide base pair is substituted for another, as you see in this example that's circled, it's called a single nucleotide polymorphism. Well, why is that so important? Because it can alter the function of the protein that is encoded by that gene. And here's a really good example of that. In the first example, gene A from person 1, we see that nucleotides, when put in triplet, GCA, is called a codon. And this codon then codes for specific amino acids. GCA codes for alanine, AGA, arginine, and so forth. In this second example, we see that in the first codon, guanine is substituted for adenine, and there's no change in that particular amino acid that is encoded by this particular codon. But in this, from person 3, if you notice in the second codon, AGA now reads AAA. And instead of arginine, now we see it's lysine. And that can change the amino acid conformation of a particular protein, leading to impairment either in a skeletal protein, an enzyme function, or how a receptor site on a cell membrane functions. So these subtle gene alterations, or SNPs, lead to virtually all chronic diseases associated with aging. And these gene susceptibilities, depending on the environmental factors, things like nutrient deficiencies, exposure to toxic chemicals or radiation, or even insufficient intake of bioactive food compounds, will then increase the risk of that particular person's disease susceptibility. So this is a nice little way to differentiate genetics versus genomics. In the case of genomics, we don't have inherited diseases. There may be an inheritance pattern that we don't know about yet, but for all intents and purposes, genomics is not about inherited diseases. There are gene mutations in genetics, but not in genomics. Single nucleotide polymorphisms exist in the genomic model, whereas in the genetic model, there are no such SNPs. What's different about genetics is that the mutations are rare, whereas in genomics, the occurrence of a SNP is greater than 1% of the frequency in population. So while it has a weak impact compared to a loss of function mutation, when these genes either are working together or not working together, they can create an additive effect, whereas in a genetic mutation, there is no such addition of how one gene impacts another. As I said, genomics is shifting away from monogenetic to polygenetic, and the last difference here is how gene expression can be modulated. In genomics, the answer is yes. In genetics, where there are mutations, there is no way to manipulate gene expression. This slide illustrates how a gene SNP starting within the nucleus can impact the cell function as well as the organ function, leading to different types of phenotypic changes, either in weight management, hormonal health, nutrient utilization, detoxification, how well a person performs in sports and athletics, whether there is a gene-drug interaction, which is pharmacogenomics, how health and wellness is impacted by a particular gene SNP, and whether a person has a risk for cardiovascular disease or diabetes can be found in the cardiometabolic health issue. Bone health can be really explored, and the underlying mechanism as to how someone has either osteopenia or osteoporosis can be explored from that mechanistic standpoint at the gene level rather than just treating things with Band-Aids. And last but not least is that we're now getting a handle on how genes influence someone's emotional health. And we'll talk about this in a few minutes. So we've covered personalized nutrition and how we want to get a better handle on a client's genotype. We've differentiated between genetics versus genomics. And now we're going to launch into how genomics is being used in medicine today. And the area of interest is going to be nutrigenomics, where we can assess and add a value to our clients or patients well beyond assessing a disease predisposition. And the first example we're going to use is transmethylation and transsulfuration. But before we do that, I want to reiterate that nutrigenomics is the study of the impact of nutrients and bioactive molecules in foods and spices on gene expression. And the research that's going on today at an exponential rate is looking at how food constituents can mitigate the effects of gene SNPs on biochemical and metabolic pathways in the prevention and or treatment of specific chronic diseases. And how do they do that? Well, these dietary bioactives function as cell signaling agents. They convey information about the diet and its components to cells and subcellular components in biological systems. These systems actually sense these dietary components at different levels, whether it's the genome, epigenome, proteome, or the metabolome. Gene and protein expression, along with metabolite production, is altered in accordance with the level of the nutrients sensed by that cell. We're going to look at folate, and this is a quick review. We're going to differentiate between folate versus folic acid. We're going to run through the signs and symptoms of folate deficiency, the causes of folate deficiency, the impact of MTHFR gene on folic acid metabolism, as well as some of the other downstream genes that may impact methyl output from the transmethylation, transsulfuration pathway. And to that end, folic acid, as you all remember, is an oxidized synthetic compound, and it's used in dietary supplements as well as in food fortification. It undergoes a reduction and methylation in the liver to form tetrahydrofolate, and the enzyme that's involved is the dihydrofolate reductase enzyme. Any excess folic acid has been linked to colon or prostate cancer, and I'm going to talk about this in a few minutes. In contrast, folate refers to the various tetrahydrofolate derivatives naturally found in food. They're metabolized to tetrahydrofolate in the small intestine mucosa, and the best sources are listed as either steamed vegetables, chicken, calf liver, as the rich sources of folate in your diet. When you have a deficiency in adults, you usually see these signs and symptoms. What's important to remember that DVT is one of those issues that we oftentimes forget, as well as impaired detoxification if you have a folic acid deficiency. In infants and children, what we usually typically see is that there's a slow growth, a weight loss, and a loss of appetite. In pregnant women, we want to guard against neural tube defects or spina bifida and miscarriage if a woman is folate deficient. There are many reasons why an individual might be folate deficient. It can be due to malabsorption, kidney dialysis, smoking, or alcoholism. It can be an inadequate intake, or there might be a B12 deficiency. We can have an excess excretion of folate, or these medications listed here actually rob the body of folate. So MTHFR is a gene located on chromosome 1. It encodes for the enzyme methylene tetrahydrofolate reductase that I'm sure all of you are familiar with. It's a pivotal enzyme in folate metabolism. It's critical for the generation of methyl groups because those methyl groups help phospholipids, amino acids associated with detoxification and the recycling of methionine. Here are some facts about MTHFR SNP and its prevalence. I see in my clinical practice about 40% of my population base having a SNP at least on one or more of these particular genes that are involved in MTHFR. When there's a reduction in the enzyme activity, it can not only lead to changes in the methylation cycle, but also in the transsulfuration pathway. The prevalence of these SNPs, like the T allele, can vary from 13 to 57% in various ethnic populations, and likewise the C allele from 17 to 42% in various ethnic populations. This is a quick overview of how folic acid or the folate cycle donates a methyl group to recycle homocysteine back to methionine. And what you see in the middle of this slide, from methionine down to sulfate, is the transsulfuration pathway. These methyl groups, as I said, are critical to the methylation of DNA. The term we now use is called epigenetics. It's important in the methylation of RNA and the methylation of neurotransmitter because that's how you degrade them, as well as with other molecules such as 2-hydroxyestrone and 4-hydroxyestrone. I hope in that book that Randy was reviewing, Tammy talked about the biochemical metabolites of estrogen metabolism, because this is another route by which women are exposed to estrogen levels that can create and predispose them towards estrogen-induced breast cancers. I like to review this methylation pathway, but I actually start at the end of the pathway, not the beginning. If you recall, methionine is biotransformed into homocysteine through many different biochemical pathways. The second step releases these CH3 groups, which are then used in various biochemical pathways. The enzyme that's necessary to convert homocysteine back to methionine is methionine synthetase. When that is combined with the next link in this pathway, which is methylcobalamin, that donates the methyl group from cobalamin to homocysteine to recreate methionine. And the last step in this pathway is how folic acid or folate participates. We have tetrahydrofolate that undergoes a biotransformation to 5,10-methylene tetrahydrofolate. But in order for the 5-methyl tetrahydrofolate to donate that methyl group to cobalamin, we need MTHFR. Let me give you a case history. This is homocysteine, which is a biomarker for a impaired MTHFR. And this is a 57-year-old Caucasian man who is pretty healthy. And interesting enough, he was taking a multivitamin and multi-mineral supplement. His diet was mostly vegetarian. He came in to see me because his father died of aortic aneurysm and kidney cancer. And while everything in his blood work appeared normal, there was a high homocysteine level of 12.2. And if you recall, the reference range is from 4 to 10. When I checked his genome and found that he was heterozygous for the 677C greater than T SNP, as well as the 1298A greater than C SNP, I recommended the 5-methyl tetrahydrofolate at 400 micrograms. Within 60 days after he had another homocysteine level check, it was 7.2. I know this slide is quite complicated, but I've tried to help you here by circling in red all of the genes that are part of the folate cycle and the methionine cycle. So you see that MTHFR down here converts the 5,10 methyl tetrahydrofolate to the 5-methyl tetrahydrofolate, donating that methyl group to cobalamin, B12, which then helps recycle the homocysteine molecule back to methionine. But as you can see, there are many other genes that are associated and creating enzymes that are involved in this pathway. So if you're focusing on MTHFR, you may overlook the fact that there are additional genes and enzymes that are critical to balancing the transmethylation transsulfuration pathway. And here I've listed the gene on the left-hand side, the name of that gene, and what the nutrigenomic intervention would be if you had a particular SNP on one or more of these particular genes. So what are the clinical pearls based on case histories generated from the Genesis Center for Personalized Health? 1, 5-methyl tetrahydrofolate supplementation may or may not lower homocysteine level depending on the gene SNPs downstream from MTHFR. What we recommend is using a genomic testing company that evaluates the genes beyond this MTHFR in the folate pathway as well as in the sulfuration pathway. We want a gene testing company that quantifies whether that gene SNP either is upregulating or downregulating enzyme activity, and we want to identify what the nutrigenomic interventions are associated with that particular gene SNP that is found in the literature. So if you are presented with a patient who has an elevated homocysteine, we like to use a holistic approach at the Genesis Center for Personalized Health, and that is to evaluate all the genes in that folate and sulfuration pathway. We want you to check the dietary intake of folate, B12, B6, choline, beta-ene, TMG, just to make sure that that individual is getting an adequate supply of these very important B-complex vitamins. Check to see if the patient has a digestion or absorption issue if they are not getting a reduction in their homocysteine level, even though you're adding all of these particular vitamins and minerals. Check all medications and make sure that the lifestyle choices of that particular patient is not robbing that person's body of folate. I want to spend just a few minutes looking again at how smoking, extreme exercise, insomnia, anxiety, inflammation, toxins, and trauma, things that we are stress that we encounter on our everyday basis, influences glutathione reductase, particularly if there is a SNP in one or more of the genes in the folic acid pathway. If stress is increased in an individual that is lacking the ability to donate a methyl group to homocysteine, then the conversion of methionine to homocysteine is reduced, which then pulls down these levels of cystathionine, cysteine, cysteine glycine, as well as glutathione, so that the net result is basically you do not have relief from oxidative stress and free radical pathology that ordinarily you would get if all of these systems are functioning adequately. Okay, so we've hit all of the high points here. I'm going to turn over the presentation to Dr. Bobby, who is going to be sharing with you something about optimizing the nutrient utilization of coenzyme Q10.
Thanks, Joe. Okay, we've had a lot of biochemistry and a lot of genomics. So let's go into something that I think you are all familiar with, and that's CoQ10. I would bet most of you either take CoQ10 or recommend CoQ10 to your patients. It's a very critical nutrient. It is found in every cell in the body, and it's involved in several different pathways in the body. I think most of us know that it is an antioxidant. What a lot of us don't know is that it also is involved in the basic production of energy, of ATP production. And while most of us get enough in our diet, stress on a daily basis from all sorts of different sources can deplete our supply. One of the most common reasons why people can become depleted in our society is the ubiquitous nature of statin drugs. And why is this? When we look at CoQ10 and where it's distributed in the body, you can see it's distributed in all the critical organs in the body. What I find fascinating is that while it declines with age, some tissues are more than others. And one question that I don't think anybody has answered yet is what comes first, a decline in CoQ10 and aging of the cells, or aging of the cells creates a decline in CoQ10? Nobody knows that yet. Now, why is CoQ10 reduced by statins? I think we're all familiar with people who have had side effects of statins. The most common is aching joints and muscles, and also pretty debilitating fatigue. And why is that? I'm going to show you a little bit of biochemistry, but what I really want to show you is what we have is a conversion of acetyl-CoA, which is involved in the basic ATP cycle, and it undergoes a complex process in which it is then formed into cholesterol. Statin drugs block the formation of this part of the pathway. What they also block is formation of CoQ10, right? So it took a long time. People finally put two and two together and started recommending coenzyme Q10 when people are taking statins. But it doesn't work for everybody, and the question is why? Ubiquinone, which is CoQ10, has to be converted into an active form called ubiquinol. Ubiquinol is the active form in the body, and it is a very strong antioxidant, and it is the primary form. Some people ask me, well, you know, if you have a choice between taking CoQ10 or ubiquinol, why don't you just take ubiquinol and not worry about it? Because it's really expensive. So the question is, do you need to spend all that money on a supplement that you may or may not need? Here's a little bit of a busy slide, but I want to show you that CoQ10 being converted into ubiquinol is actually part of a larger system of antioxidants. If you see, it's also looking at vitamin E and vitamin C as part of that cycle. So it's really critical. Antioxidants, as you know, scavenge all the free radicals, which are what cause DNA damage, causes premature aging, and most of the chronic diseases. So where does genomics come into this? We have an enzyme called NADPH, or quinone oxidoreductase, which is coded by the gene called NQO1. If you have a SNP on NQO1, you have a much greater risk of having a ubiquinol deficiency. Ubiquinol, remember, is the active form. And because ubiquinol helps protect your DNA, if you do not have enough, it increases the risk of many of these diseases, including cancer. So why do you need to know somebody's gene type? Why can't you just say, well, just give them ubiquinol or just give them CoQ10 and just measure it and see if you're doing right? Most of you are familiar with the ability to measure CoQ10. There's different labs that do this, and as you can see on this one, this person has a normal level of CoQ10. So we're all set, right? Well, if you don't know that this person has a gene SNP on NQ01 and can't convert that CoQ10 into the active form ubiquinol, you are being misled as a practitioner and misleading your client that they're actually well stocked with ubiquinol. Okay, ubiquinol is really a difficult chemical to measure in the bloodstream. It's very unstable, and so last I checked, no commercial labs are measuring it. So if the only thing you can measure is CoQ10 and you don't know what their genes are doing, then you don't really know whether this person is actually getting what they need, and that's the ubiquinol. Okay. Another really common area for people these days, and it's a very hot topic, is vitamin D metabolism. And I'll go through that and show you how your genes can really impact what you recommend in terms of vitamin D. Here's a little basic overview. All right. We all know that the main source of vitamin D for many people is sunlight. That's why in the northern latitudes where the sunlight is less strong, we can sometimes have an issue with insufficient vitamin D. So sunlight gets converted through several different pathways into vitamin D, and the liver and the kidneys are main organs involved in this process. Most people think of vitamin D as being involved in bone health and regulating calcium. It's actually it's a hormone, and it is involved in cell signaling and talking to other cells and other genes, and so can impact a variety of pathways and disease conditions, including autoimmune disorders, cardiovascular disease, depression, anxiety, and even things like dementia and Alzheimer's. So it impacts a lot of different systems in the body, which is why when somebody's deficient, it can really wreak havoc. Here's a look at various genes along the entire pathway. Here you have sunlight being converted into vitamin D, and the gene isn't on here, but it's DHCR7. So right away you have to have a gene coding for the enzyme to get the vitamin D. GC codes for the protein that carries it to the liver, where it's then hydroxylated to 25-hydroxyvitamin D, and then it's carried into the cells and converted into 1,25-dihydroxyvitamin D. What's really important is this form, the 1,25-dihydroxyvitamin D, is what actually gets seen by the receptors on the cell and they bring that vitamin D inside the cell, where it then starts talking to other genes and other systems in a very complex cell signaling system that you can see here. And this, by the way, also involves vitamin A, which is one of the reasons why vitamin A is important in bone health. Okay. So as you can see, any of these steps along the way, if these enzymes are not working properly or the receptors not working properly because there's a gene SNP that may impact that function, that can potentially cause low levels of vitamin D or low levels of vitamin D in this cell that are actually functioning. Here's a slightly different look at what happens. You have DHCR7, as I said, converting the sunlight into cholecalciferol, and then you have the conversion into the 25-hydroxyvitamin D, and that's regulated by CYP2R1. And then you have GC, which carries the vitamin to the cells, and then you have several vitamin D receptors that bring that vitamin D into the cell where it can actually do its work. Right? So any step along the way can impair the body's ability to either produce or utilize the vitamin D. And the more SNPs you have, the greater the potential impairment. So what's the take-home lesson from this? We all know to check vitamin D levels, right? That's part of our routine evaluation. Most people only check the 25-hydroxyvitamin D. But as you can see, the 125 dihydroxyvitamin D is the one that actually gets into the cell. And so there are multiple enzymes along the way, and you need to measure both of these forms of vitamin D in order to get an accurate picture of what's going on for that person, right? So if you have a difficulty converting 25-hydroxy into 1,25-hydroxy dihydroxy, you may actually have normal vitamin D according to standard labs, but you may actually be deficient in the very form that you need to get into the cell. All right, and based on your levels of these two forms, then you can decide, does this person need extra supplementation? How much do they need? What form do they need? And one of the things that my patients always, don't like to hear, that their sunbathing actually has no benefit in terms of their vitamin D. So these are all important in working with your clients in terms of guiding them to how much do they need of this vitamin D, and how do you assess whether they're actually getting what they need. So now I'm going to turn this back over to Dr. Joe, and he's going to talk about mental and emotional health.
Thank you, Dr. Bobby. Great presentation on vitamin D and coenzyme Q10. We're going to shift gears right now and talk about mental health or emotional health. And what I did was try to put a word map up to describe some of the things that people relate to when we talk about mental health. It is one of our increasing health issues in this country, evidenced by the number of deaths occurring recently on college and high school campuses. One of the things that has been hopeful in this regard is that we're finding out that there are certain genes that play a role in someone's mental and emotional health. And what does it mean to have a positive emotional or mental health? It means to function in society, meeting the demands of everyday life, and to recover effectively from either an illness, change, or misfortune. And there are specific genes that we now know influence neurotransmitters in our brain. Catechol-methyltransferase is one of these. A SNP on COMT is linked to an excessive level of these neurotransmitters, L-DOPA, epinephrine, and norepinephrine, which can result in anxiety, bipolar disorder, adult onset alcoholism, hypervigilance, just to name a few. And this is personal, because the case history that I'm citing here is me. I didn't know this until I did my own genomic test, but I have a SNP on COMT. It's actually homozygous, meaning that it's the worst possible case, meaning that the COMT does not effectively degrade these neurotransmitters. And then I started to put two and two together. I said, oh, this may explain why when I'm driving and I'm hemmed in by lots of other cars, I experience anxiety and hypervigilance. And I did a little experiment on myself, because SAMe at 400 milligrams per day has been recommended in the literature to act as a catalyst to speed the degradation of these neurotransmitters. Guess what? I found that I had decreased anxiety and hypervigilance when I was driving in my car, but I was hemmed in by other vehicles. Some other genes that are taking prominence right now in the emotional and mental health area is the dopamine receptor. A SNP impairs the function of this receptor, meaning that it does not see dopamine circulating in the bloodstream. And a paper published in 2011 by Van Rokel showed that a SNP on DRD2 is associated with adolescent loneliness, especially when there is little or no parental support. This is the gene-environment interaction. Remember, these gene SNPs predispose us, but if these genes are put in a positive environment, then we don't get a negative outcome. The same thing is can be said for the brain-derived neurotrophic factor, BDNF. This encodes for a protein responsible for neuronal circuitry, a fancy word for neuroplasticity, or your ability to deal with situations or events that come up in your life. When you have a SNP on BDNF, it's linked to all those things that we have been talking about: mood disorders, anxiety, and depression. And a paper published in 2014 showed that adolescent girls who are carriers of this homozygous allele for BDNF scored higher for loneliness than boys. And this is where gene-gender interactions occur. And when we do our, we are a genomic testing company, and so our reports help practitioners understand that there are gender differences in how a particular person responds to one or more of these SNPs. And before closing, I want to talk about drama and trauma, and how epigenetic changes associated with this BDNF gene can influence not only this generation, but subsequent generations. This was done, this particular study was done with rodents, and what they did was create a very stressful environment for and actually they were maltreated. And what happened was that the females passed on a DNA methylation on BDNF to their offspring. And those offspring were unable to care for their young. How does this relate to what's going on today in our culture? Early abuse and neglect creates a transgenerational change in the gene expression, not in the DNA code, but through epigenetic markers on the genes, and create behavior changes not only in the F1 generation, but in the F2 generation. So we've covered all of the high points in the learning objectives tonight. Who benefits from functional genomic testing? I list quite a few here. Dr. Bobby talked about the nutritional paradox, where nutrients, which I consider pronutrients, have to undergo a biotransformation before they're metabolically active. It's important for competitive athletes or weekend warriors who are looking for an edge to know what their genes are telling them about either nutrient utilization or what the best exercise is for them. And we know that today based on more than 20 years of research in the literature. How does it benefit you or your client? Well, first of all, it's a blueprint. It helps you guide that client in getting the best information to improve vitality and longevity. It helps you create a comprehensive and effective treatment plan based on the backbone of functional nutrition. And because it's increasing in popularity and acceptance, you're going to be known as an innovator. And it keeps your clients and you on the cutting edge of this rapidly growing science and technology. So genomics is part of the omics revolution. It adds value for your clients well beyond assessing disease predispositions. It sustains relationships and adds value, and the patient gets a better outcome. And more importantly, it personalizes treatment therapies and strategies. It eliminates what I call the OSFA, the one-size-fits-all, and it provides that blueprint with actionable lifestyle steps for you and your client to work on together. We've provided some online resources A good one is actually the U.S. government's very interested in the human genome. I provided this 1000genomes.org website, which is this latest information that I shared with you earlier in this presentation. We have, at genomicsolutionsnow.com, educational webinars in these categories, Introduction to Nutrigenomics, and for those that are not really familiar with genetic concepts, we have a primer for those that need to get their arms wrapped around these concepts again so that they understand nutrigenomics. We also have our own blogs, and Ty has his blog, which he calls Dogomics, and he was just talking about oxytocin. And there was a paper published that when a person looks into the eyes of a dog registers an increase in oxytocin, which is your feel-good hormone. And guess what? It works it works in the opposite way too. So when a dog looks into your eyes, you also register an increase in that hormone called oxytocin. Ty's final thought, he said, cats don't do this. Here's some genomic textbooks that you can use as references and resources. In 2016, we are projected to publish through CRC Press a book on DNA testing and nutrigenomics from the clinician's point of view in the prevention and treatment of chronic diseases. If you want to contact either one of us, you can do that through our general email address at info@genomicsolutionsnow.com. And if you're interested in looking at some of the test panels that we offer to clinicians, you can check us out at our website, www.genomicsolutionsnow.com.
So
go ahead.
That concludes the presentation part, and we would love to hear your questions. This is our favorite part.
Yes, go for it.
All right. Thank you so much for that overview and comprehensive explanation. So we'll get to some questions here. First, I want to let everybody know that this is being recorded, and the full recording and slides will be up on our website in just a few days. And so you're talking about B12, and do you think so you agree that the methyl form of B12 is the most effective form to take as a supplement?
Absolutely. Yes, without a doubt. It provides, if there is a impairment in that person's ability, and particularly in the folate pathway, to donate that methyl group to the B12 molecule, it's an insurance policy, as I like to say.
Let's see if I understand this question. If you take methyl B12, does that decrease the amount of MTHF needed?
No.
No, they're two separate parts of the pathway, so they're both just as important.
Yeah, they're actually, in a sense, they're compartmentalized, and for someone who has an MTHFR SNP, that's associated with the conversion of 5,10 methylene tetrahydrofolate to 5-methyl tetrahydrofolate, whereas B12 and the gene that is involved there is FUT2, is involved in the intrinsic factor that is necessary for the absorption of B12 from the GI tract.
Okay. On this same topic, the MTHF donates a methyl group to the B12, but if B12 already has a methyl group, what happens?
It's all about moving those methyl groups around. So they're all donating and receiving, and it's the cycle in which they do that. So it may.
Yeah, you want to keep that cycle always moving, because remember, those methyl groups donate to the transsulfuration pathway, and a impairment in that particular function will lead to less glutathione and increase your free radical pathology.
Mm-hmm. Okay. If you could provide the names of the activated forms of the B vitamins.
I think what the 5-methyl
Mm-hmm.
tetrahydrofolate is the well, let me go back. You okay with me going back, Paula?
Sure.
Okay. I'm going to go back to that slide because it lists the nutrients. There it is. So it lists the nutrients. So if you have a SNP on MTHFR, you want 5-methylfolate.
Mm-hmm.
If you have a SNP on FUT, you want a B12 or methylated B12. MTR, same thing, methylated B12. Methionine synthase reductase, again, because this is involved in the cobalamin transfer of the methyl group to the transsulfuration pathway. You want a methyl group on that B12. B6 is necessary for C-beta S, or synthethionine beta synthase. And B12 is also necessary, the methylated form, is for TCN2. Last but not least, and we often forget this, is that there is a particular gene called MTHFD, methylene tetrahydrofolate dehydrogenase. And the nutrigenomic intervention here is choline. Oftentimes we think if we supplement with the active forms of all of the B-complex vitamins that we've got ourselves covered. If you still have an elevated homocysteine level, it's probably due to the methylene tetrahydrofolate dehydrogenase gene SNP. And again, if you're working with a good genomic testing company, they're evaluating all these. And that's what we do in our nutrient utilization genomic test.
Okay, thanks. While we're on this, trying to group questions together, does an individual also need adenosine B12 or only the methyl? From my understanding, they need both.
My experience most of the time is the methyl works fine. And remember, this is all about balance. I think what I've seen with people who tend to get into trouble, it's because they're out of balance. And people sometimes think more is better. And what you have to remember that everything in the body is a yin and a yang. And so everything you do has a consequence. And it's not a matter of trying to, super dose people. It's a matter of trying to find the balance. And this is where using both the genomic testing as well as your biomarker testing can really help you in that. The other thing to remember is life is not just about the folate pathway. And if your only focus is on this, you tend to get myopic and you forget about all the other pathways that play with the folate transsulfuration pathways, and you may be losing sight of other things that may be impacting. That's why it's always best to take a broader view.
Okay, great. So someone has been taking methyl B12 and methylfolate and tests high for serum B12, folate, and B6, but still has high homocysteine. What do you suggest next?
You're missing a few genes.
Yeah.
Go shopping. Go gene shopping.
Yep.
Yeah.
Yep. You made the assumption that it was an MTHFR or a B12 issue, and it's probably one or more of these other genes listed here or circled here in red that are contributing to the elevation in homocysteine.
And remember the impact of stress. Okay, So stress will negatively impact this cycle as well.
Okay, thanks. Sorry, balancing this.
Yeah.
And then they move. I read that hydroxy B12 is best. Since methyl is too exciting, I'm not sure the question, but methyl B12 doesn't bother them. So again, I think that goes back to the methyl and the hydroxy question.
Yeah, and again, what I recommend to people is don't get so focused on one particular thing that you lose sight of the whole picture, and that's what I usually see happening. And so if you're having an issue with some of the different supplements, take a step back. What else is going on? What's the bigger picture? What am I missing? What am I forgetting? And not focusing on one or two pieces to the exclusion of everything else.
Well, this is an interesting point that's not come up before. This person's webinar panel screen is over the slide that's up. And so you can put your cursor on the top of it and move that screen, but let's ask the question for you. The screen's covering up the intervention for the gene TPX. So is MTHFR vitamin B1 or B12?
Oh, B12.
Thank you. And is there a methylated form of choline, and is that needed?
No, it's not.
Okay, some vitamin D questions. If a person puts liquid vitamin D on their skin, will it be absorbed?
That's a good question, and I think it depends on the formulation. Our skin certainly is a great vehicle for absorbing a lot of different things. And just as if you put it in their body under their tongue, the test of it is measuring how much actually gets into their system.
Thank you. And then when there's high vitamin B12, would that indicate a backup someplace?
A high vitamin
B25D, yeah.
In the serum or in the red blood cell?
I'm not sure.
Okay. I mean, if you're looking at vitamin B12 in the serum, if it's high, it's most likely because they're taking a lot. It generally is cleared fairly quickly. If you want to know how a person's utilizing it, that's when you start looking at the biochemical pathway and the different components and looking at how much is actually getting into the cell in the first place.
Okay.
One thing to remember with labs is always remember, what are you measuring? Mm-hmm. We'd love to measure everything in the cell because that's what we're concerned about. That's really hard technically. So we're always taking a proxy measurement and extrapolating, and sometimes that works better than others.
Yeah. Yeah, again, it's a follow-up question. I'm not sure if it's serum or red blood cell because the concern is it going to be back up into the kidney? So seems, well, I would say serum, but anyway.
Yeah, it more than likely is serum. I have not seen a vitamin B12 toxicity associated with the kidney.
Okay. So this person is homozygous MTHFR C677T. So it's a topic that they're very much interested in. They were listening to Richard Koonin, MD, on recently, and he mentioned that betaine TMG, which is a very inexpensive nutrient, he says can bypass this genetic pathway. He didn't go into much detail, but it caught their interest, and wondering if you can elaborate.
Right here. Well, yes, this is another route where homocysteine can get routed. If you can see my cursor, you've got choline, betaine, and then you've got this enzyme, BHMT. It's a methyltransferase enzyme that can be that can have a SNP on it. So if that gene is functioning and the enzyme is functioning, you can again circumvent all of this left-hand side of this particular curve. However, a lot of the functioning of MTHFR and folate is associated with pyrimidine synthesis as well as neurotransmitters. So it's important to have this system functioning and not rely on an alternate pathway whereby you can recycle methionine at the expense of this folate cycle.
So again, to me, this is about becoming a little myopic. You know, if you're focused on the homocysteine part of the cycle and reducing it, yes, that's where the betaine can come in. But there is a whole other function. The folate cycle is not just about generating methyl groups. That's what we talk about, and that's what a big part of the focus is. It has a lot of other functions in terms of, as Joe said, the pyrimidines and purines. This is, you know, basic building blocks. So it has a lot of critical functions that if you're not moving things through that pathway, you're missing a lot, and your body will eventually show that.
And I also would like to point out, just as a sidebar, an elevated homocysteine can be associated with a SNP on CBS, or cystathionine beta synthase. If this gene that encodes for this particular enzyme is impaired, you're going to get a buildup of homocysteine. Likewise, if you are serine deficient, you could get a buildup of homocysteine. And it may not have anything to do with the choline BH methyltransferase enzyme.
So a couple of things you could clarify on the homocysteine ranges here. This is what the person said. So I've seen that range saying that less than 15 was normal for homocysteine. This person says if your homocysteine level is at 4, it is high, and what steps do you mention to take?
A homocysteine level of 4 in terms of standard measurements is not high.
It's not an excess.
Mm-mm.
So I'm not sure what the question is.
Maybe the person left out a 1.
Maybe. So the first question is like, is 15 less than 15 normal? Or is that high?
Okay. This is where you get into standard lab interpretation versus functional lab interpretation. Yeah. Okay. And those of us who practice functional nutrition and functional medicine know that just because a lab says it's within a standard value range, that doesn't mean that's actually in the ideal functional range. And the ideal functional range may vary from person to person. You have to remember that the lab standards are based on deficiency and excess, really relating to disease. You know, they're not focused on what actually might be an optimal range or an optimal level for optimal function. Vitamin B12 is a perfect example. You know, most labs say you're not deficient until you're below 200 or 300. But then they'll notate that you can get neurological symptoms at, you know, 400 or 500. So again, it's knowing what are you measuring and how do you put that into a context.
Okay. There was a point where you were talking about exercise. Questions, what do you consider extreme exercise?
Oh, in response to the stress on the and the resulting increase in free radical pathology. Good question. Everyone is different. Everyone is unique. And what we try to do, because we have a sports and exercise panel, is to try to match what your genes are telling us with the form of exercise that is appropriate for you.
And the other, just to add on to that, not only what form, but the intensity. We look at the inflammatory components, which can increase risk for injury. So some people, overtraining for one person may be fine for another. And then obviously in nutrition. So again, it varies from individual to individual what constitutes overtraining or overexercising and what constitutes actually an optimal level. You can get into the emotional aspects too. You know, exercise can be an addiction as well, and that's a whole other discussion.
Okay.
But we have genes
looking at that
in our emotional and mental health panel that will give us a clue as to whether someone is predisposed towards either an eating behavior addiction or other types of addictions that exist today.
Mm-hmm. Well, here's a good one. As nutrition professionals, nutrition coaches, are we able to order genomic testing through your company?
Absolutely.
And have to try it for themselves first. Is that possible?
We recommend you do it on yourself first. That is the best way to learn. It becomes a personal experience, and your learning curve is greatly shortened. And from our perspective, nutritional professionals are the perfect professionals, the perfect area to be doing this, because you guys understand the power of food, you understand the importance of food, and this is a beautiful tool to help you just add to your personalized approach.
And could you speak to what the reports look like? Do you have a copy of one that you can show? Are you going to show that next presentation?
That's in the next presentation.
Okay.
Just to give you, I'll give you a basic overview. Our reports have basically three main sections. The first section is a bird's eye view of what's going on with the genes. They're color-coded so that all the reds are grouped together, which are the high impact, all the yellows are grouped together, which are the moderate impact, and then the greens, which are little or no impact, and what areas those are in, as well as the genes. So very quickly, at one glance, you can see where somebody has a lot of high impact or moderate impact genes in a pathway and where you need to focus. Obviously, you need to overlay that on their clinical picture. The next section we actually include, because most of us can't remember all the functions of the genes, especially as you get into the bigger panels. So we include a description of the normal gene function for every gene in that panel. So it's a great reference, and if you're talking to clients, it's easy to be able to refer back to that rather than having to remember all that. And then the last section goes into the actual results. And you know, Joe and I are both clinicians, and we design these reports for clinicians. We're all busy, we don't have a lot of time, so the results give you not only what the potential impact is for that person, it also gives you the action steps, what has been shown in the literature and that we use in clinical practice in terms of diet, lifestyle, exercise, nutritional supplements, to be helpful in mitigating the potential impact of that disease. And what are the biomarkers? What are the measurements that you can do to know that this intervention is working the way it's intended? Rather than waiting 10 or 15 years to see if somebody gets a disease, you can actually take a peek into the biochemical process and see what is going on and do I need to adjust.
Okay. So you're very much speaking to this question of what's the role of the dietitian or the nutrition professionals here. So
critical.
Anything you want to add to that, right?
Yeah. Yeah, because everything that we recommend are things that are in your stable. These are your areas of expertise. You know, dietitians and nutritionists all have training in lifestyle coaching, in diet, and nutrition, and even supplements when they're needed. You know, they're called supplements for a reason. That the diet is the basis of everything, and then supplements are only needed to add on when you can't get what's needed in the diet.
Mm-hmm. So there's a question about pricing for the test. I know there's a variety of tests, and so go to their website, but is there a place that they need to look?
Yeah, to access the pricing, you just have to register as a healthcare practitioner, and once you are approved, you can have full access to the practitioner side of things, which includes detailed descriptions of the panels, the genes, and the pricing.
Yeah, there's a lot of information on the website. It's a good thing to review. And you provided a number of resources. People are asking about a list of the websites and the books that you mentioned. So those are on their slides, and the slides will be posted to our website in just a few days. So you'll have access to that. And I see Joe scrolling here, so we're going to rest on that slide, you know, right here.
Yeah, we also have several hundred articles on the website, on our website.
Mm-hmm. Yes, really good ones.
Yeah, we have a library that healthcare professionals can access. We're updating our website, and there will be a searchable feature associated with that library, so that if somebody has a keyword, it will pull up the articles pertinent to those keywords.
Mm-hmm. Makes it easier to find what you're looking for. So hopefully that'll be up in a few weeks.
Okay. Another good question here is, when you're doing the test, do you recommend people stop supplements for any length of time before testing, or if nutrients have been taken a long time and supported a weak or lacking gene that could possibly influence the outcome?
No, your genes are your genes. And
no, remember one thing. Genes change expression of a particular protein. They don't change. SNPs don't change. Once you have them, you got them for life.
Yeah. So no matter what you're doing, it does not impact the actual genes. So it doesn't influence the test.
Nutrients can influence gene expression, but not the gene itself. It can't change a SNP back to a normal gene.
Okay. I think it's probably time to put the elephant question on the table. So how does your testing differ from a 23andMe SNP test?
That's a common question we get a lot. I break it down into a couple different parts. One is we are a clinical testing lab, and so the lab is certified from a clinical standpoint and has to follow various guidelines clinically, which 23andMe does not. It's a higher standard. And also the security of your data. We don't sell your data, 23andMe does. They've made billions of dollars selling data to big pharma, and they're now using the data to develop their own drugs. And then obviously 23andMe doesn't have reports. There's no interpretation. They test for a lot of genes that nobody knows what they do or how to intervene, and our criteria for testing gene SNPs are that they have to be validated in the scientific literature. What is the function? What is the potential impact on somebody's health? And just as importantly, is there an intervention or interventions supported in the literature that can change that outcome? Mm-hmm. And those are the criteria that we use.
Yeah, and that's really big.
And just as a segue and a plug for the next webinar that we're about to give in a couple weeks, there is a slide in that presentation comparing Genomic Solutions Now, the panels that we have created, versus 23andMe.
Yeah. Okay. So it's comparing, you know, it's not really apples to apples, it's apples to oranges. And if you are a researcher and you love to do research, and it doesn't, clinical literally doesn't matter as much to you, 23andMe is fine. I will tell you that Dr. Joe and I have spent thousands of hours in the literature researching, and we do it on a daily basis to keep up with what's going on.
Right. And with 23andMe, it's like you say, you know, there's no information, there's no reports, no source to evaluate. But I mean, that's the thing with them. You've got to go to an outside source to to get it interpreted.
And Paula, I'll just share this. There are some people that can interpret and understand research papers and come away with the essence of what that article says. There are others, however, that can misinterpret what a researcher has found or the conclusions that they have identified in that particular research. It's really, I'm getting to the point where it's really, really important that you have a lot of confidence and trust in the people that are doing the grunt work, if you will, which is basically going into the literature and reading these research articles and coming away with the action step or the information that is going to be the most useful for patients or clients.
Great. All right, thank you. I'm looking forward to next presentation. You know, we'll get into more case studies. I think really seeing how what the reports look like and you know how they're used with individuals, and just what your clinical experience will be a very, very interesting presentation. So I'm looking forward to that.
Yeah.
Great. We have appreciated the questions. We know that nutrition is evolving, and it's moving more towards the biochemistry, particularly as it relates to genomics. And we appreciate your students and alumni taking all of this in and digesting it all.
That's right. Absolutely. Well, thank you. And just a reminder, the date for this part 2 of this presentation on with Dr. Veltmann and Dr. Kline is on November 17th. And again, we'll start at 3:50 Pacific time with our Food News You Can Use segment, and we'll start the webinar at 4 o'clock. And again, this was recorded. It'll be up on our website in just a few days, so hold on to your hats. It won't be that long. It'll be good to review this again. There's a lot of information, right? And a reminder that there's a survey to fill out after the webinar ends. Let us know your feedback so we can do our best to serve you every time. All right, everybody. Thanks so much, and we'll see you back on the 17th.
Have a great night.
Thank you.